Hi everyone. Today we're going to have a webinar based on a recent publication by members of the STAR team. We will run in the usual kind of way. We'll have the presentation and then we'll open it up for questions afterwards. So please feel free to use the chat for any questions or discussion points, and we'll follow them up at the end of the presentation. Today we got two speakers, so it will be headed up by Aki Heikkinen, and he is a principal scientist in SimSip. He's in the translational science team, but he has extensive experience with academia, pharma, and more recently was in a CRO business before joining us in the translational science team. And of course, in that time period, he's gained a lot of experience, particularly on PBK, IVE, and in vitro methods focused on transporters, metabolism, and DDI, and is part of the team working towards both development and evaluation of transporter and metabolism mediated DDI qualification activities and verification. Ivan has joined us more recently. He was doing a master's previously with Amin's group in the University of Manchester, and he part of this work that will be presented is related to this, which was continued on after his master's. The topic today is one of these things that comes round every once in a while, discussions around static models versus dynamic models. And this paper looked at several areas, topics, and they will cover these in the presentation, but particularly looking at the comparison of these static and dynamic models, looking at competitive inhibition in both hepatic and intestinal metabolism, some critical kind of analysis looking visually how the space of DDI predictions between static and dynamic models, and also raising the important consideration looking at vulnerable versus average individuals. So I'll pass over to, I believe, Aki first. Thank you for agreeing to present to us today.
Yeah, thanks, Ollie. Okay, so yeah, I will start firstly, like, probably majority of you know that just a bit of a background on why we are interested in drug interactions. So unanticipated, unrecognized, or mismanaged interactions can be a problem and can cause patient morbidity or even mortality. And therefore, understanding how to manage interactions is important and may allow safe use of interacting drugs. And the questions we are interested in there is, the first is that does a drug alter the PK or PD of other drugs, or the other way around that does other drugs alter the PK or PD of the investigational drug? Thereafter, obviously, we are interested in the magnitude of any of these interactions, and also that whether the interaction has clinical significance or not. And in case the interaction is clinically significant and relevant, then what might be the appropriate management and prevention approaches for those to avoid the unwanted outcomes of using. Sorry, there is some background noise coming from someone. Okay, and then as there are so many drugs in the market, and so it's impossible to test clinically all the possible scenarios. So we need to predict this. And with any method, understanding the strengths and limitations of these methods is needed for their rational use. And for metabolic interactions, two of the key methods for doing these predictions are mechanistic static models and dynamic models. We are addressing in this this webinar and in the publication we worked on. So just briefly, that mechanistic static models, the substrate and inhibitor concentrations do not change over time, so it's static, and but different concentrations are used for different organs. And when we are talking about cytochrome P450 metabolism, then the relevant organs would be liver and gut. And the mechanistic static model suggested in the regulatory DDI guidance is expected to result in conservative predictions, and it's really geared towards over-predicting the DDI risk rather than under-predicting it, or that is the expectation. And with dynamic models, the substrate and inhibitor concentrations change with time in different tissues in the body, but here relevant for these interactions within intestine and liver. And just some features that can be addressed with dynamic models, but not with mechanistic models, would be that, for instance, effects of formed metabolites and transporters can be considered. Auto-inhibition, auto-induction can be considered as well. Mutual interactions between the substrate and inhibitor can be factored in dynamic models, and that would be a challenge with mechanistic static models. And also impact of various study design effects like dose staggering, changing formulations, splitting doses can be factored in. And relevant for the work and the presentation here, one key point is that interindividual variability in the characteristics and mechanisms affecting both substrate and inhibitor concentrations and the interactions between them can be considered in dynamic models more readily and more in detail than what would be achievable with mechanistic static models. So, but even though there are several applications where mechanistic static model clearly cannot replace dynamic models, there have been recently in the literature during the recent couple of years papers addressing the questions that can mechanistic static models be used in place of dynamic models, and more specifically that for which applications mechanistic static models could be used in place of dynamic models, and also asking that what driver concentration should be used for the inhibitor in the static models. And we decided to look into this and see if we can come up with any applications where we would be comfortable with the suggestion that the mechanistic static model would be replacing dynamic models. And just mention a few of those papers that have been addressing these questions from some angle. So there are a couple of papers from Pfizer, if I'm not mistaken, addressing time-dependent inhibition of CYP3A4, and then in another paper, other P450 enzymes comparing static and dynamic models. In those papers, they compared average individual predictions with observed clinical data. and also explored the differences on different assumptions on driver concentrations and what should be used for the inhibitor to improve the how well the clinical data is captured. And another paper on from Boehringer Ingelheim, I believe, was looking at if the mechanistic static models for DDI could be used to support regulatory filing and study waivers. And also in that case, they looked in the average individual predictions and compared it with observed clinical data. And just briefly summarizing the results in all of these, that in some cases, mechanistic static models for the average individual can result in similar predictions than dynamic simulations. However, in all of these, they showed also discrepancies between these models, and in general, the dynamic model predictions were able to capture the clinical data better than the mechanistic static models. The discussion and conclusions in these papers varied a bit, and the points of view and the conclusions they wanted to convey were slightly different. But these are how I would interpret the results they present. So then going into our work. So this is just a screenshot of the title of the paper where we published this work. So what we looked at, competitive inhibition of metabolism in both gut and liver. So difference to the work by Pfizer in the previous slide. They looked at MBI, mechanism-based or time-dependent inhibition, whereas we are just looking at competitive inhibition. Instead of looking at real compound pairs and clinical data, we run just a simulation study comparing the static and dynamic model predictions. With this, that we are not bound to scenarios and drugs for which we have clinical data, we can cover wider, while realistic parameter spaces for conceivable interaction scenarios. Well, the potential drawback of that approach is that we don't have a direct evaluation against clinical data. So it's comparison of the static and dynamic model predictions. And the question we wanted to address was that does mechanistic static model reproduce dynamic model predictions like the papers I referred to in the previous slide suggested that in some cases it is possible. And maybe more importantly, that if we could actually foresee that just based on the mechanistic static model predictions, that whether this we would expect to have a discrepancy between these models or not. So then going into the mechanistic static model, a bit of background and trying to explain where it's coming from. Even though I believe that for many of you, these models are you're familiar with this, but just some of the key assumptions included in the mechanistic static model. Well, if we go first to this equation, so it's predicting AUC ratio. So AUC, oral AUC of the substrate in the presence of inhibitor or inducer over the AUC without interaction. And that is split into two components, the interaction in the gut wall and in the liver. And so there it's the fraction escaping the gut wall in the presence of inhibitor over the fraction escaping the gut wall metabolism without interaction, and the intrinsic clearance in the liver without interaction over the intrinsic clearance in presence of interaction. And this is actually quite important that it's oral administration of substrate, and it's not only related to the gut wall metabolism, but also the liver part is assuming oral administration. If the substrate would be IV administered, this equation would not be applicable. Well-stirred liver model is assumed here, underlying there, and also as we are talking about intrinsic clearances, non-saturable elimination of the substrate is assumed. And as said before, the static perpetrator or inhibitor concentrations in the gut and liver are assumed, but the concentration, the static concentration estimate in the gut wall and liver, they differ from each other. Yeah. And something not relevant for the current presentation, but just to mention that the interactions altering the enzyme levels, so meaning mechanism-based inhibition and induction, they basically, the model assumes that they are in steady state as well. So it's not only the exact time when the interacting compound is either inducing or inactivating, but so that the turnover of the enzymes is at steady state the new levels of the object or enzymes. So only competitive inhibition was accounted for in the current work, and these equations are just the component that is competitive inhibition is included in here. And we had several scenarios simulated there. So when the substrate Fg, so the fraction escaping the gut wall, is 1, only interaction in the liver has an impact on AUC ratio, because this the part covering the interaction in the gut wall, that will be 1 when Fg is 1. And there are just some references on the background of the mechanistic static model and the assumptions underlying these models. And then going into the perpetrator concentration side of the mechanistic static model. So the concentration in the gut that is derived from the fraction absorbed, the absorption rate constant, dose, and then the blood flow clearing the compound from the gut wall to circulation. And with this, we are basically assuming that the inhibitor present in the circulation does not contribute on inhibition of the enzymes in the gut wall at all. So even though the compound would be building up, accumulating after repeated dosing, the mechanistic static model actually assumes that that will not increase the inhibition in the gut wall, and that is something relevant for the discussion later on. Then the maximum unbound hepatic inlet concentration. So this is the equation used in the regulatory guidance, and that is expected to represent the worst case scenario or the highest possible concentration of the inhibitor in the liver. We did run all of the all the comparisons with this version of the mechanistic static model, but and that is included in the paper, but I will not cover those in detail in this presentation. We will focus on the average unbound hepatic inlet concentration, where the liver concentration is predicted otherwise the same equation, but it's the average steady state concentration in circulation that is driving the interaction, and then the inlet concentration that is coming from a single dose given at the same time with the substrate. And in order to minimize any bias in the model comparisons, the blood flows both to the gut wall and liver, they were matched with the Simcyp healthy volunteer population representative. They are slightly different than the values suggested in the regulatory guidance, but this was just to minimize the bias between in the comparisons. And the Cmax and the average concentration at steady state, they were taken from the corresponding dynamic simulation results to allow a fair comparison of these models. And also all the other parameters, the input parameters which went into the models were the same for the comparisons. For dynamics and mechanistic static model predictions using the same input parameters. Then the dynamic model, what we used, the Simcyp Simulator version 21 was used. The substrate first order absorption model was used there. So that is the parameters going in there is the absorption rate constant and the fraction absorbed, lag time also, but that was set to 0 in all of the cases, both substrate and inhibitor. And the first pass metabolism in the gut wall is predicted with the QGut model within the simulator. Minimal PBPK model and well-stirred liver is used there. And a single dose of substrate was administered at the same time with the inhibitor at the time when inhibitor was at steady state. So for the inhibitor, first order absorption model was used. So that model is deriving the driving concentration for inhibition in the gut wall that is estimated from the portal vein concentration. So that is the portal vein concentration times the fraction pound in gut. And that also points out that the portal vein concentration will be dependent on any accumulation of the inhibitor in body after repeated dosing. Minimal PBPK model, well-stirred liver also for the inhibitor, and in this case for the liver, unbound concentration in the liver is driving the inhibition for hepatic enzymes. And as mentioned earlier, repeated dosing until steady state before administration of the substrate, and then also repeated dosing was continued until the end of simulation, and the simulations were running long enough to see the substrate profiles, full profiles, also in the inhibited state. And as we covered a wide range of parameter space, there were low clearance compounds and with high volume of distribution. So it had to run the simulations fairly long after the administration to cover the full AUCs, both with and without interaction. And with that, I will hand over to Ivan to cover for the simulation study design and some of the data visualization.
Thanks, Aki. So yeah, I'll get started on explaining how we carried out our comparison of the DDI prediction methods. So as mentioned, in general, the aim was to explore if static and dynamic models are equivalent in predicting metabolic DDIs, and specifically arising from competitive CYP inhibition of a single substrate by a single inhibitor. And this is the area we want to focus on because it's the simplest DDI scenario, so it's quite easy to explore. And this is where discourse remains within the industry as to whether the models are equivalent in predicting DDIs, specifically when using the average concentration at steady state in the static model. So the gist of it is we would simulate a DDI within Simcyp, which is our dynamic model, and then use identical input values from our dynamic model within the static mechanistic model, and then compare the AUC ratios we get from both models. And this gives us a direct comparison of how the static and dynamic model predict the same interaction. And so we wanted to do this on a very large scale to capture the range of drug parameter spaces that are theoretically possible and see what effect that had on discrepancies between the models. And the way we did this is that we have selected parameters of interest that we thought could contribute to discrepancies between models, and we had 7 of these parameters to which we assigned a range of values. We would then vary these parameter values independently across all possible permutations, allowing us to generate a large number of distinct drug-drug interactions. And this is described by the schematic on the right-hand side, where the top level sim represents our overall simulation, or our overall DDI, and then each subsequent level represents parameters and values that we assigned for said parameter. And this pattern continues for all Explorer parameters, and the respective values are represented by parameter X. So each simulation result is generated from the various combinations of different values for each parameter, which gave us exactly 30,000 distinct drug-drug interactions. And obviously, this would have taken a very long time to do by hand. So we used the SimSip R package to automate the process. So the changing of the parameter values within SimSip, running of simulations, input values to static models, and the comparison of the dynamic and static models pretty much was all automated. And just to mention that the majority of our simulations were run with PopRep to simulate the average individual, but we also ran simulations with a population of 300 individuals and then taken the 95th percentile AUC ratio from the population to mimic a vulnerable individual, and then compared these results to the static model. Next slide, please. So we did not, so the compound that we used for our study, we did not generate from the ground up. Instead, we used templates. So for the substrate and inhibitor, we used midazolam and ketoconazole as templates respectively. And we picked midazolam and ketoconazole because midazolam is primarily metabolized by CYP3A4 and ketoconazole inhibits CYP3A4, which allowed us to explore the impact of intestinal metabolism on discrepancies between models. And as Aki said, we use minimal PBK with first order absorptions to basically minimize the difference between models and to maximize comparability. On the left-hand side, all the parameters you see have been kept static throughout all of our, were kept constant throughout all of our simulations, and then parameters on the right are our parameters of interest in which we assigned a range of values and varied across our simulations. For the substrate, we looked into the fraction-escaping gut metabolism and the fraction-escaping hepatic metabolism. And for the inhibitor, we looked at the volume distribution in steady states, the absorption rate constant, fraction of drug-unbound enterocytes, the CYP3A4 inhibition constant, as well as the dosing regimen. And as Aki said, all of these values were kept realistic, so we could actually compare these results to the real world, even though we don't have distinct clinical DDI studies for a direct comparison, but we wanted everything to be physiologically plausible. Now, for the inhibitor, changing the parameters was quite easy since we had direct inputs within SimSip for all of these values. But for the substrates FG and FH, we do not have direct inputs, so we had to use a roundabout way of changing these values. For the fraction-escaping hepatic metabolism, the change in this value was achieved by either decreasing or increasing both the maximum number of metabolism and the Michaelis-Menten constant for the CYP3A4 route of elimination. And for the FG, instead of changing compound parameters, we actually varied the CYP3A4 enzyme abundance in the intestine for the population. And while we could have used or changed parameters in midazolam such as QGUT or FEGUT to change the FG, this was likely to change other parameters, which we would have to account for. So instead, we went with changing the CYP3A4 enzyme abundance for the population in the intestine. And this also did not affect the FG of the inhibitor, which was good, so we did not have to account for that. Next slide, please. So the main metric that we used to kind of describe our discrepancies was this intermodel discrepancy ratio, or the IMDR, which is simply the AUC ratio we got from the dynamic model divided by the AUC ratio we got from the static model. And this IMDR value was then categorized based on discrepancy intervals, where an IMDR of in between 0.8 and 1.25 meant no discrepancy, and an IMDR of above 1.25 indicated patient risk discrepancy, because in this case, the AUC ratio of the static model was significantly lower than the AUC ratio of the dynamic model, which means that perhaps in a real-world scenario, just using a static mechanistic model may have missed this interaction, which is associated with patient risk. And an IMDR of below 0.8 indicated sponsor risk, because in this case, the AUC ratio from the static model is significantly higher than the AUC ratio from the dynamic model. And so this could perhaps, in a real world scenario, lead to a false positive result, leading to unnecessary clinical DDI studies, which poses a cost to sponsors. And so the way we visually presented our results is through this plot on the right-hand side, where on the x and z-axis we have two parameters. So on the x-axis we have the absorption rate constant for the inhibitor, and the values increase from left to right. And then on the z-axis we have the inhibition constant for CYP3A4, and the values increase from right to left. And then it's important to note, so our main figures are made up of 25 of these individual plots. And in these individual plots, the x and z-axis always stay the same. So it's always, yes, as shown here. So the x and z-axis always stay the same. And the y-axis shows our IMDR values, which are color-coded based on the discrepancy intervals. So the green zone shows that there's no discrepancies. The red zone is associated with the patient risk discrepancy, and the yellow zone is associated with the sponsor risk discrepancy. And so as we can see in our main figure, the individual plots stay the same with the same x and y and z axis, but as we move between the plots, left, right, up, down, we change the values for inhibitor VSS and substrate FG. So as we move upwards with each individual plot, we're increasing inhibitor volume of distribution at steady state, which ranges from 0.05 to 31.25. And as we move from left to right between the plots, we increase the substrate fraction-skipping gut metabolism. And obviously we have three more parameters which we want to look at, but since we're running out of dimensions and axes to show these on, the inhibitor dosing regimen, substrate FH, and inhibitor FU gut can only be compared in between figures. And so each row that you see on the right-hand side table here represents a separate figure. So if you wanted to compare, for example, dosing regimen, you would have to basically find the corresponding substrate FH and inhibitor FU gut values, and then just compare the figures with different dosing regimens. And that's all I have to say. Thank you.
Okay, thanks, Ivan. I will continue on the results. So first, going through the average individual comparisons, and just this is giving a summary table here, not going through all of these, but just pointing some observations from here, and then going into individual cases from of this. So both patient risk and sponsor risk cases were observed, and so in total 66% of the simulations were in the sponsor risk range, and about 1% of the scenarios were in the patient risk. So clearly showing that big part of the or majority of the predictions do not fall in this range where the predictions can be considered equivalent. And all of these are presented as figures in the paper and the supplement of it, and so you can go and look at those. But I will cover the few cases to make the points for the questions we want to make with this this work. So first, looking at the case where the inhibitor dosing regimen is once a day, 400 milligrams. There is considerable first pass of the substrate in the liver, so high clearance compound, and the inhibitor FU gut is low, so the dynamic simulations will predict fairly low inhibition in the gut wall. And here, if we look at first the right-hand side, so the cases where the substrate FG is 1. So these figures, the interaction is contributed only by interaction in the liver, and the gut doesn't really play any role here. And there we see actually a small range of patient risk cases, and so these scenarios are associated with scenarios where the inhibitor volume of distribution is small, and with that a peak to trough ratio of the inhibitor will be fairly high. And so there it is possible that actually the dynamic simulation predicts higher interaction risk than the mechanistic static model. Whereas as the inhibitor concentration in the gut wall in the dynamic simulations is the free fraction is low, the sponsor risk cases will dominate in the scenarios where there is substantial gut wall metabolism of the substrate. Then this one, otherwise similar case, but the inhibitor FU cut is 1. So assuming that more inhibition in the gut wall for the dynamic simulations. The right-hand side, where gut wall doesn't contribute at all, this is identical to the previous slide I showed. But there are, when we go to cases where gut wall metabolism is playing a role, we end up seeing scenarios where actually the dynamic simulation is predicting more interaction than the static predictions. Whereas overall the discrepancies are towards the sponsor risk, and so the yellow areas are dominating in over the red areas here. Then comparing this again, otherwise the similar case, but just changing the inhibitor dosing regimen from QD 400 milligrams to BID 200 milligrams. So the daily dose and thus the average concentration at steady state in circulation will be similar, but the peak to trough ratio in the dynamic simulations will be different, and also the concentration predicted for the gut wall will be different in these. So what happens there is that as the peak to trough ratio is more narrow with BID dosing, there is a better match between the static and dynamic models for the hepatic interaction than with the QD dosing. But still there are scenarios where the yellow areas appear. Whereas with the gut wall metabolism, actually the red areas where we are predicting more interaction for the with the dynamic model, those areas will be more prominent than in the QD dosing scenario. And what is contributing on this, that why we are predicting more interaction with the dynamic model than with the static model, especially in the gut wall, can be demonstrated with the predicted concentrations driving the interaction in the gut wall. As I mentioned earlier, the static model is actually predicting the gut wall inhibitor concentration just from the dose and absorption rate and fraction absorbed, and it's not taking into account that accumulation of the inhibitor in the body could actually increase the inhibitor concentration in the gut wall. Whereas with the dynamic simulation, with the model we have used here, the driving concentration for inhibitor in the gut wall is derived from the portal vein concentration, and that is increasing, and it can actually be higher than in these simulations than what you would predict with the static model, and that is explaining why we predict higher interaction in the gut wall with the dynamic simulation than with the static model. Interactions in the gut wall, that is notoriously difficult to get spot on, and we don't really have good case examples to how accurate these predictions are, but this is just illustrating that building of the or accumulation of the inhibitor in the body could actually contribute on interactions in the gut walls as well. And then looking at one scenario where the inhibitor dosing regimen was PID 200 milligrams, lower clearance substrate and less first pass metabolism in the liver for the substrate, and again limited inhibition in the gut wall due to binding. And when we look at the just the average individual comparisons, the sponsor risk scenarios dominate here, and some of the predictions end up in the similar range. But when we look at the vulnerable individuals, so the dynamic models, we take into account the population variability and look at the 95th percentile. So the most vulnerable individuals in this is with healthy volunteer population simulations, 300 individuals. So there we see actually several cases where the dynamic simulation and the vulnerable individual is the DDI risk is predicted to be substantially higher than with the for the average individual in the with the dynamic simulation, but also with the static model. And just illustrating that looking into just the average individuals, and that may not be sufficient to actually cover the interaction risks that are realistic in the population. So with that, I just list the conclusions on. So the mechanistic static model total, it doesn't reproduce the dynamic model predictions for an average individual. So 67% of the simulated scenarios here were outside of the 0.8, 1.25 range. And the sponsor risk is more prominent, so about 66% of the scenarios were there, and the patient risk about 1% were in that range, but both do exist. And overall, it may be really challenging to foresee if there will be a discrepancy without actually running the dynamic model simulations. And then if you are considering a scenario where you would like to use the static model for decision making, and so it actually may be really challenging to conclude that if dynamic model would be actually useful or not, and or well, to put it the other way around, if the mechanistic static model would be sufficient, or would dynamic model be actually required. So you would actually need the dynamic simulations to be run to conclude that. And the patient risk is associated with scenarios where the substrate has high first pass metabolism, either in the gut or the liver, or both. And for the substrate with high first pass in the liver, but not in the gut, the patient risk is associated with scenarios where the inhibitor has a low volume of distribution. And the predictions of average individuals neglect the vulnerable individuals with higher interaction risk, and these individuals are likely to exist in real patient populations and should be taken into account in the evaluations. And with that, I'm happy to take any questions and comments.
Thanks, Aki and Ivan. Yeah, if there are any questions, please take the opportunity to raise your hand now, or write a message in the chat. Hi, Nora.
Hi, thanks a lot for the presentation. Maybe I missed it, but in your summary, you were talking about all the situations, or kind of that it's difficult to say when this will apply, but did you identify any scenarios where the static model could be applied?
Oh, well, sorry. Well, yeah, we do see the green areas here, but it's yeah, it will be really challenging to see the foresee if your compound is actually lying in this these areas. So because well, we are it's a limited number of parameters we are covering here. It's still not covering all possible scenarios, and so yes, this could be used for guiding that if you think the predictions could be similar, but I would think it's actually you may get it may be easier with just build the dynamic models and run the simulation there, and but then trying to use the mechanistic static model for the quantitative DDI prediction. If you have the dynamic simulation already and you don't, it's not really useful to use that mechanistic static model for that prediction anymore.
Thanks.
Any other questions?
I saw someone else raising their hand, but
Yeah,
lowered it again. So
I think they maybe raised it by accident. I'm not sure. Okay, so if there are no further questions, we're actually on a bit of a theme looking at interactions. This time we're going to be hearing from Luke in May on the disconnect between 3A4 time-dependent inhibition. So please feel free to register for that. That'll be on the members area registration. I think as well, just to follow up as that Aki will be at Marbach Castle as well, just in a discussion with this topic as well. So I think that'll be a useful discussion point as well. Thank you everyone for joining today, and yeah, take care.